The second portion of photosynthesis is the light-independent reactions, also known as the dark reactions or the Calvin cycle. Occurring in the stroma, the Calvin cycle builds glucose using CO2 from the air, a molecule called rubisco, and the products from the light-dependent reactions, ATP and NADPH. The Calvin cycle can be broken down into 3 stages. Let's take a look at the first. The first stage of the Calvin cycle is carbon fixation, named because carbon being harnessed from the CO2 in the air will serve as the backbone for the glucose molecules, the final product of the Calvin cycle. Here, we see a select area inside the stroma of the chloroplast. In brown, we have rubisco, short for ribulose biphosphate carboxylase, which binds with CO2 to temporarily form a 6-carbon molecule. Because it is unstable, this 6-carbon molecule quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. To keep things simple, the diagram only shows the carbon atoms and not the other atoms in the molecule. The enzyme phosphoglycerate kinase, shown here in pink, then moves the phosphate group from an ATP molecule to a single 3-carbon molecule. Now, let's go on to stage 2, reduction. The enzyme glyceraldehyde 3-phosphate dehydrogenase reduces the charge of our phosphorylated 3-carbon molecule by removing a hydrogen ion from NADPH. It then releases this hydrogen ion and moves the electron to our phosphorylated 3-carbon molecule. So, one could also say that NADPH is oxidized to become NADP+. What we now have is a molecule called glyceraldehyde 3-phosphate, which is abbreviated G3P. Now, it's important to remember that along the way, we have broken down a larger carbon-containing molecule into several smaller ones, and ATP and NADPH have been used for each 3-carbon molecule. Thus, 2 ATP are used for each molecule of CO2 that has been fixed, and the same goes for NADPH. Also note that lots of reactions are occurring simultaneously in the stroma, so many G3P are being created at once. Of these various G3Ps being produced, some will leave the cycle and some will stay. The third stage of the Calvin cycle is regeneration of rubisco to be used for carbon fixation. Basically, for each of the 12 G3Ps produced, or 6 CO2s fixated, 2 leave and 10 G3Ps remain to be transformed back into 6 rubiscos. The G3Ps that leave will be used to build a glucose molecule, which stores energy originally captured as light energy for later use.